I'd like to thank the organisers for putting together this conference and also for giving me the opportunity to talk. Today I'm going to talk to you about applying hypergraphs to the modelling or study of biological systems. I'd like to introduce my co-workers. They are Samuel Barton, the University of Queensland, Adele Costa, who is at the UNSW, and James Lefebvre, who's also at the University of Queensland. I'm going to begin by first of all introducing one of the data sets we were looking at. So, Senecio is a plant that grows in various locations around Australia. In particular, it grows on headlands down the eastern seaboard and also in the sand dunes close to the sea. But this plant exhibits very different growth patterns depending on the location where it is sited. And the data set is the result of an experiment used to try and find a link between the genotype and the phenotype of the Senecio plants. So, seedlings were gathered from both of these types of location. There was a control group for each of the gravitropic and agravitropic species, and a treatment was applied to each of these groups. The treatment was to turn the plants on their side and see if the growth pattern returned to normal. Then measurements of gene expression using RNA-seq were taken at 5 distinct time steps and the results recorded. Now normally you would study this in using maybe a graph or a network where the vertices or nodes were the genes and there was an edge connecting every pair of genes, with the edge weighted by the correlation between the expression levels for each of the genes. But in this particular experiment, we were working with gene sequences, and there were over 200,000 of these gene sequences. So the resulting graph is very dense. You can use techniques like clustering and other things to try and distill information from the graph, but we were looking at taking an alternate approach. So essentially, we were trying to identify significant perturbation in the expression levels for around 269,000 gene sequences. We were only interested in those that were significant, and so we identified those genes for which there was a 2-fold change in expression levels for, say, from one time period to the next. And in addition, to try and take out noise within the data, we only included those results where the expression or the difference in the expression level satisfied a p-value of less than or equal to 0.5. So this distilled down our graph significantly, but again, we've still got over 200,000 vertices in the graph, and it's quite dense. as an alternate approach, we're looking to capture not only the pairwise interactions between the genes, but the multi-way interactions. And in particular, when there was significant changes across multi-way comparisons. So on the left here, you have a standard graph representation where the vertices are the genes and the edges represent some sort of comparison between two genes. A hypergraph takes an alternate approach. So here the vertices or the nodes are aligned with conditions. So it might be, is there a twofold change from time period 2 to time period 3? That's a condition and that's a vertice. And the edges now do not just link two vertices, but they collect together a number of vertices which in this which satisfy these conditions. So a hyperedge is an edge containing a number of vertices. The hyperedge is a gene, and each gene corresponds to a hyperedge, and we include a vertex if that gene shows significant perturbation with respect to that condition. And in this way, we capture extra information about what changes or what perturbations are relevant to each individual gene. Taking this information, we can translate it into mathematical structures and then study them. But again, what we have is still a dense graph, because now the number of hyperedges is over 200,000. But by using mathematical techniques, we can collapse down this information and represent it more succinctly. And that's what the representation on the right is supposed to indicate. So here's an example from the data we had. And you can see that in the middle, we have actually a representation of a hypergraph. Here the purple dots are genes, and you can see that the information here identifies a set of 8 genes. 2 genes that behave very similarly to almost all other genes, and another 6 genes which just have similar perturbation patterns as each other. So the hypergraph can identify individual genes or sets of genes that behave differently. Still, we would like to know more of what's happening in this dense matter, and so we can distill the information down even further, and that's what the representations on the left and the right are telling us. Now, given all of this, we can use standard graph theory techniques, such as the study of centrality measures, which identify central vertices or nodes, which are genes here, which behave in a particular way, or are central when compared to the behaviour of the other genes. And that's what's going on here. And we can see that these centrality measures, in fact, partition up our gene set into 4 distinct parts. And it actually, whilst it still looks somewhat dense, it does identify individual sets of genes, small numbers of genes, which behave differently and distinctly from the rest. Using this information, we can go back to the biological data, identify the gene sequence which corresponded to that node or gene, map it against known annotations, and map it against their functions. So we could then distill the information we have and show that this technique was identifying genes which were central to some sort of transportation within these plants. Before I finish, I want to briefly tell you about a second study we were looking at, and this was, we had a library, a reference library for plant microfossils across 7 distinct species, and we were taking ungraded starch grains, for which there was a total of 944, and using the reference library to allocate or classify these starch grains to their species. This gave us a hypergraph classifier which used length, area, perimeter, circularity, island position, and 11 mathematical coefficients to help us identify the species for a given starch grain. Once we finished, we calibrated this classifier against a standard random forest classifier, and in the graph on the left we can see that the hypergraph classifier performed at times better than the random forest classifier, justifying that this is a reasonable approach to take to classify distinct species or classes. We also looked at the sensitivity to each of the parameters. So just wrapping up, hypergraphs are flexible models applicable to diverse biological settings. Hypergraph models can allow us to study multi-way interactions. This allows us to reduce biases and overfitting, and also to incorporate what's called a population of models which improved results. Further work in this area would be to understand the parameter setting within the models, and also the feature selection through probability distribution. So thank you for listening today, and I look forward to answering your questions.